for a second. Yeah, welcome everyone to our March seminar. It's my great pleasure to introduce Suvrat Kotagal. Actually, I did not ask you how to say your name correctly.
Fine.
Did
Suvrat Kotagal, yes.
Suvrat Kotagal. Okay, so I
That's better
didn't
than
know.
many people pronounce it in India.
Okay, good. Yeah, he already said it. He is originally from India, but currently doing his PhD together with Gudrun Kadereit at the University of Munich. And that's also where I met Suvrat, I think 2 years ago, more or less, in the summer when I was visiting Munich. And he was the person who knew every melastome in the greenhouse there and knew exactly whether it was flowering, whether it flowered the whole year or not, or was fruiting. And how long the flowers are open for most species. So I was very impressed. Suvrat told me yesterday that he actually didn't really know Melastomataceae while he was still living in India. He saw an Osbeckia once in the field and only later realized that it was actually a melastome. And his deep passion for melastomes came in Munich in a class when he started working with Melastomataceae. And yeah, as so many of us, I guess he once fell in love with them and then sort of got stuck. So
Yeah.
probably he has a very long career ahead because Suvrat is addressing topics in Melastomataceae that, as far as I know, no one else is working on. So I think you have a big treasure box in front of you to dig into. So yeah, you can go ahead and share your screen, and we're all excited to learn about the heteranthry and the genetics that underlie complex flowers.
Thanks, Agnes, for that nice introduction. So, can you all see my screen now?
Yeah, perfect.
Or are you also seeing the?
I'm seeing
I
the
hope.
second slide now, the introduction slide.
Okay. Is it? Okay.
Now I'm seeing the start, yes.
Okay. Ish. Come on. Yeah. So let's start. So okay. So I will be talking about staminal dimorphism and mostly focusing on the genus Arthrostemma today. And it's because we chose it as a model species for the beginning of our studies, and it will be mostly focused on this genus, but I will also talk about other stuff that I found interesting in the greenhouse. And the talk will be mostly in two parts. The first one will be looking at the transcriptomics and to see what candidate genes we found, which are responsible for this dimorphism of stamen. And the second part will be more focusing on the function of this dimorphism and future interesting things that are possibly on the way. So let's start. I don't need to introduce melastomes to anyone here. Everyone knows it's a huge family and it's mostly buzz pollinated with these pollen rewarding flowers. And there's a huge variation in staminal morphology that's seen in this family, which has been linked to pollinator specialization and also diversification in the family. This staminal traits, especially the pedoconnective and the connective appendages, are highly labile. Even within closely related taxa, they can be uh, very different, and sometimes even within the same species, you can see differences in morphology between the appendages. And as I will show you later, sometimes the same species has isomorphic and dimorphic forms as well. And one of these staminal traits is stamen dimorphism, as you know already, that this is a very prevalent character in melastomes. It's the different kinds of stamen or two different morphs of stamen occurring in the same flower. It's also called heteranthry. The differences can be with the size of the stamen, color, connective appendages, the scent. Some can produce nectar, some cannot, and all this is you can read in the books, and many research has been done already. But this trait is also highly labile. Across the whole family, we can see multiple origins and reversals of this trait of stamen dimorphism, as I've marked here in this. Let me have a quick pointer. Yeah, so I've marked here from this phylogeny. And we see multiple origins and reversals of this trait, and this high lability of this trait led us to ask the question, is there a simple genetic basis behind this that is causing this evolutionary lability, which might also prove to be a valuable insight into how the family diversified in terms of species as well as stamen morphologies. So we wanted to do this with the transcriptomic approach, and for this we need live material. So I went into the greenhouse, looked at all the melastomes we had, what time they were flowering, what kind of stamen morphology they had, scored everything and made a huge table, and was looking for closely related species which had different morphologies, and it ended up that we had two species of Arthrostemma, which one of them, which is Arthrostemma parvifolium here, which has iso-sub-isomorphic stamen, and another one, Arthrostemma ciliatum, which has clearly dimorphic stamen. And they were in the same genus, so closely related species having different morphologies was a very interesting thing for us, and we chose it as the model species for our studies. So just an introduction to the genus Arthrostemma. It is in the tribe Rexiae along with two other genera, Rexia and Pachyloma. It has 4 species. It is distributed only in the neotropics, and it's also invasive, I think, in Hawaii and also Brazil, but still in the neotropics. So it's also has invasive potential elsewhere, but so far it has not been classified as an invasive. And this genus is interesting because it has 4 currently recognized species. Doing a literature search, I found, and 2 of those, the parvifolium and alatum, have iso-sub-isomorphic stamen. One of them, Arthrostemma primavum, has dimorphic stamen, completely dimorphic, and this Arthrostemma ciliatum has 2 different populations, one of which has dimorphic stamen, one of which has isomorphic stamen. And there is also by geographic distribution of these populations if you look at it in the literature, but I will show you later. It's not very clear. And this Arthrostemma ciliatum, I didn't know it when I started it, but it's proving to be a really, really interesting model species for this study. That's why this Arthrostemma is the 0.5 species here. So just stamen morphology of Arthrostemma ciliatum, it has, or at least the morph that we have in the greenhouse here, it's a dimorphic form, and it has these anticephalous stamen with the long pedoconnective, which are supposedly, if you are looking at the normal theory for stamen dimorphism, which is pollen or functional separation between the 2 different morphs. This is supposed to be the pollinating whorl, outer whorl, which has longer and which are camouflaged against the petals, and the antipetalous whorl, the inner whorl, are supposed to be the feeding whorl for the bees. And to do the transcriptomic comparisons, I looked at the development of the flower because we also wanted to see what stages is this dimorphism actually, how is this dimorphism actually over the development of the flower. So I tried to identify different stages which would be interesting for the comparison of using transcriptomics, and I ended up with 4 different stages. The first one with the very small flower bud where both the whorls look more or less the same. The second one, which is slightly larger flower bud, where the outer whorl of stamen is only starting to gain its pedoconnective and the appendages, while the inner whorl is not showing any sign of the initiation of the appendages. The next stage, where the outer whorl is right in the middle and the dimorphism is very apparent in this stage, the outer whorl, the appendages, and the pedoconnective are in the middle of development, while the inner whorl is also showing slight signs here of the of the appendage. And the last stage, which is the more or less the completely developed stage just before the flower opens, where you can see the morphology like when the flower is open. So we wanted to extract RNA from each of these stages and from each of these worlds by separating the worlds. So I dissected each of these stages, developmental stages, separated the two worlds, outer and inner worlds of stamen, and then we extracted RNA from all of these to do comparisons at each developmental stage between the worlds and also between developmental stages within the same world. So we extracted RNA, sequenced it, assembled the transcriptome, I put it through quantification in Salmon, and then I performed differential expression analysis. So the first step of the differential expression analysis is data exploration, where you give the transcript abundance to it and see how are the expression profiles of your different samples. So as I outline here, the outer stamen are always marked O and the inner stamen are marked I, and the different stages are marked with their different numbers here. So the developmental stage 5, 3, 18, and 12 were all clustering together in terms of expression pattern. As we would expect, the stamen at each developmental stage are supposed to have similar expression levels. But we didn't see a very clear separation between the two worlds at each stage. As you can see, the closest, this is a sample distance heat map. So all stage 5 stamen are very close in their expression profiles, but you don't see here all the outer worlds of stamen are close to each other than the inner worlds of stamen. And this is also evident in the MDS plot that I did using the same data. So each developmental stage clusters together, but each world within the same developmental stage doesn't cluster. As you can see here, there are two inner worlds, one outer world which is clustering together. So it's not very clear, the expression level patterns, if you look at it in the beginning. But the good thing is that the different developmental stages are clustering together, and this means that the experimental design is actually working. And I went ahead and did the differential expression analysis at the transcript level, because we didn't want to do it at the gene level since we don't have a closely related genome. So I annotated the differentially expressed genes using a program called Mercator, and we did the comparisons of between worlds, that is outer world and inner world. I will mostly show you the comparisons between worlds here. And then I did functional filtering of these transcripts that we annotated to only look for genes that are involved in development of the stamen, because those genes are the most probable genes which are causing the differences in morphology. So for the first comparison, we did the outer world versus inner world at stage 3. And this is always a relative comparison. So I always use the inner world as the benchmark to compare the outer world to. So in the outer world, you can see here compared to the inner world, we found some interesting candidates which are involved in stamen development. The MIB class transcription factors, the auxin influx transporters, which is a phytohormone, which is also very essential for development. As you all know, another called EPFL precursor polypeptides, another one called DELA proteins. And in the outer wall of stamen, the MIB class transcription factors were upregulated compared to the inner wall, and the DELA proteins were downregulated. And I will, in a little while, I will tell you what these different names mean. The second comparison that we did was the next stage, and we found similar kind of transcription factors or transcripts also differentially expressed between the two worlds here. One of them is the JAS-TPR transcriptional repressor, which is also involved in stamen development. Another, again, auxin genes, related genes, gibberellin related genes, and again, this EPF precursor polypeptide. And in Arabidopsis, because it is the only, or not the only one, it is the most well-studied model here. they have shown that these genes, the DELA, as I said before, the auxin genes and the MIB transcription factors are responsible for stamen development. So they have an interaction. It's a biochemical pathway of production and also of signaling, where they signal signal and affect development of the stamen. And they are known to affect stamen development in the early stage as well as in the late stage. And these EPFL polypeptides are especially known to control stamen elongation in Arabidopsis. So these are ligands which bind to receptors in the cell and affect cell division, which makes the filament grow. But in this case, in Arthrostemma, it is actually the pedoconnective which grows longer and not the filament. So we think that this pathway of signaling might be involved with the DELA proteins, the MIB transcription factors are potential candidates for causing these differential morphology of the stamen between the two whorls. And this is also another candidate, the EPFL peptides, which might be causing the differences in stamen morphologies. And again, we went to comparing the other whorls, other developmental stages as well. We find the same genes which are involved in this pathway that I showed before, the AP2ERF ethylene response factors, transcripts. Another interesting thing was the S-locus gene, which is mostly involved in heterostyle, which has been shown in Primula and other heterostylous species, which we found as differentially expressed here. Another interesting one was a vacuolar processing enzyme, which is mostly involved in programmed cell death. So we think at this stage, this vacuolar processing enzyme is downregulated in the outer whorl, which means it is upregulated in the inner whorl. So we think that this programmed cell death might be causing the reduced stamen morphology of the reduced complexity of the appendage here. And here it is downregulated, so there is no programmed cell death, so they are free to grow as they want, and that's what is contributing to the elongation of the pedoconnective and more complexity of the appendage as well. And in the last stage, when the stamen are almost completely developed, we see genes upregulated in the outer whorl, which are mostly involved in flower opening and organ boundary domain, organ boundary determining genes, as well as gravity sensing genes. As you know, the stamen in Arthrostemma and in most melastomes are usually zygomorphic. This is only happening during the flower opening or just 1 or 2 days before the flower opens. The stamen actually move towards or away from gravity. So we think these genes might be involved in that gravitropic or anti-gravitropic movement of stamen, and more genes which are regulating flower opening and that kind of stuff. So as a conclusion to this part of my talk, we found several interesting candidates which are putatively involved in causing the stamen dimorphism in Arthrostemma ciliatum, the dimorphic form. This GID1 DELA MIB complex, which is regulating the stamen development, might be involved in elongation, differential elongation of the two stamen whorls. These were differentially expressed, and we think this is a potential candidate to look for in other species. EPFL peptide genes might also be causing this, as I said before, like by affecting differential by affecting differential cell division within the two stamen whorls, or vacuolar processing enzymes causing programmed cell death, which might be causing the reduced complexity in the inner stamen whorl, or a combination of all of these, along with other transcription factors which are phytohormone regulators. And one final candidate is the S-locus gene, which might be involved, or which might be a homolog of this might be causing this differences in stamen morphology, or it might be a remnant of a diastyle mechanism which was in this species. So for future in this direction of identifying the genes, what I want to do is also study the development at a cellular level of the stamen so that we know whether it is the cell elongation that is causing this elongation of the pedoconnective, or whether it is actually division of the cells that's causing these, to further narrow down these candidate genes into a smaller group. And then we also want to get an annotated genome of this, or one of the annotated genomes from this genus, from one of the species. And I also want to do a whole genome network analysis, not a whole genome, but correlation network analysis of this transcriptome data that we have to identify hub genes, to identify the main transcription factors, which are the central regulators of this mechanism of stamen elongation. And also we want to expand this study to other species. Like I said, we also wanted to compare the Arthrostemma parvifolium, the one which has all isomorphic or sub-isomorphic stamen. But because of flowering time periods and also lack of time for the master thesis, we weren't able to do it. But I'm planning to do it in this year to add this data set to the existing data set as well. And also we also want to look at other species in many different types of melastomes to see whether these genes are homologous, in the sense that it's the same genes causing this stamen dimorphism across the different tribes, or whether it's different genes completely. Because even the stamen dimorphism is not homologous in the melastomes, because sometimes you see it, like in Rinchantera as an example, in the same world there is dimorphism, but in most other melastome species, there's dimorphism occurs always between the two stamen whorls. And then the last step would be to narrow down and validate these candidate genes by performing knockdowns, whether we can force dimorphic flowers to become isomorphic, let's say. Now we want to, as I said, we want to do this for the other species as well, and work is underway for this with the help of students, and also I'm working on this. I've already identified the different stages that we want to compare in this species, Arthrostemma parvifolium, which has sub-isomorphic stamen, let's say. And other species which we want to also look at is Heterocentron muricatum, which the stages here were identified by a student, Anna Schlick, who was working here in the group for a while, just for a research course. And it's not just the genes that control stamen, these appendages and the morphology of the stamen. We also have to take into consideration the developmental constraints that are involved in the development of the stamen, because the pattern of initiation of organs makes quite a big difference. Hang on a second, we'll cut this. Okay. The pattern of initiation of stamen of organ primordia makes quite a big difference for this. And in most melastomes, as we know now, that it is the stamen that are developing last in the flower. So first she initiates the sepals, then comes the petals, then comes the gynoecium, and only the last initiation of organ is the stamen. So the stamens are under a lot of developmental constraints because of just space reasons while developing. And also, since this ovary, because they are folded in the bud, there's different amounts of space for them to grow in between the hypanthial wall and the ovary. And this is illustrated here. We had a lot of questions during my presentation about the developmental considerations. So we ended up doing some manipulation experiments with the flowers to see how the stamen would develop without a constraint, let's say. So we removed the petals of the flower when the flower was still in a very young bud stage, and saw how the stamen developed. So this is a normal Heterocentron muricatum stamen. You can see the appendage is quite nice and straight, but when we removed the most extreme case here, I'm showing the appendage went haywire. So this is an indication that the presence of constraints in the form of flower petals and sepals and other organs during development affects the size and also shape of the appendages a lot. And the most common change that I saw while doing these manipulation experiments was an elongation of this appendage, which was quite a bit longer than for the unmanipulated stamen. And in another case, in arthrostemma, usually arthrostemma has a very tiny knob-like dorsal appendage. But when we actually remove the stamen or the petals while development, you can see here it grew quite big. So there's not only the genetic considerations that we have to keep in mind, there's also the developmental constraints. And this will be a very important thing to consider while in going forward in the future. And much study has already been done in this field already from the developmental people in the group. So now I want to move on to the functional aspects of heteranthery in this morph of Arthrostemma ciliatum. So I told you already, according to the division of labor hypothesis, which is the most commonly accepted hypothesis to explain stamen dimorphism, if we apply it to this case in Arthrostemma ciliatum, the longer stamen would be the pollinating stamen of the outer whorl, and the inner whorl stamen, which have these yellow tips, would be for attracting the bees, which also makes sense when you consider the color of it. And these would be the feeding stamen. But Arthrostemma ciliatum is, I think, a kind of unique case in this respect, that the stigma is not close to the pollinating stamen. So this posed an interesting question for us, and it doesn't grow long either. Especially at least in this dimorphic form. So this posed a question whether this division of labor hypothesis actually made sense for this species. And so I went and wanted to study this, also with the help of the student Anna Schlick, to study this by looking at the pollen, basically the number of pollen, the size of pollen, and the viability of the pollen between the two stamen whorls. So the pollen number we wanted to count from each, we wanted to estimate the number of pollen from each anther from each whorl. So we wanted to do this by correcting for the difference in size, because you can see already here the anther lobes are already quite different in size. So of course this will have more pollen than this, but we wanted to correct this by using a size correction, or doing it by per volume area or per volume of sporogenous tissue. So we measured the diameter, the area, cross-sectional area of the anther and the length, and we correlated it, which we checked for differences between the two stamen whorls, and it turns out the cross-sectional area is not significantly different between the two stamen whorls. So we ended up using the length of the stamen as a proxy to correct for the differences in sizes. And we crushed these pre-anthetic stamen just to make sure that all the pollen were still inside. Dispersed it into a known volume of liquid, water in this case, and counted the number of stamen, number of pollen using a Neubauer cell counting chamber, which is basically a slide which has marks like this on it. And it has a standard formula which lets you calculate the number of cells per microliter. And from this, we extrapolated the number of cells per sporogenous tissue per length in this case. And you can see here, we ended up with a weird bimodal distribution in both the stamen. And we did a comparison test anyway even with the bimodal distribution, and found that it was significantly different, the number of pollen between the two stamen whorls. The yellow ones being the antisepalous whorl here, and the pink being the antisepalous one, the outer whorl and the inner whorl. The yellow supposedly being the pollinating ones, and the pink, inverse. Yellow being the feeding ones, and the pink being the pollinating ones. And even disregarding this statistical test, you can still see the distribution is not completely overlapping, and the antipetalous ones usually have more pollen. This kind of makes sense because it might be investing more for feeding instead of pollinating. Then we went ahead to check for the other two categories, pollen size and viability. The size we checked was just the diameter of the pollen, and we found that it was significantly different between the two stamen whorls. The antipetalous stamens had a significantly smaller diameter or smaller sized pollen than the antisepalous ones, which is supposedly the pollinating stamen. And this kind of also makes sense. The pollinating stamen need to have more energy to produce the pollen tube, so they might be bigger and these might be smaller. But when we tested the viability of pollen, it came out completely inverse of what was expected. The antisepalous stamen had less viability. The supposed pollinating stamen had less viability than the feeding stamen, supposed feeding stamen. And this leads back to the position of the stigma, like I said before. Okay, so this leads back to the position of the stigma, because the stigma is very close to the feeding stamen instead of the pollinating stamen. And this might be causing this difference in viability of the pollen, as the flower might be more adapting or starting to adapt to this shorter stigma and adapting a selfing mechanism, so that the inner wall of stamen function as both pollinating and feeding stamen. Or it might also be that it is starting to transition into a state of this Bertollonia-like pollen shower selfing mechanism, which might also be the case, as we see two different stamen morphs within the same species here. So this pollen tube shower selfing mechanism, like I said here, I didn't see actually pollen tubes connecting the anther and the stigma here, but in the other species of Arthrostemma that I looked at, Arthrostemma parvifolium, I actually saw pollen tubes connecting the anther or the pore and the stigma. The pollen were actually growing within the pollen tubes were growing within the anthers without any extra addition of medium, and they were just connecting to the stigma here, affecting self-pollination. You can see a picture here stained with aniline blue, the pollen tubes within the anther going haywire. And I also saw a huge, insane amount of seed set and fruit set in the greenhouses. This photograph is behind glass, so there is no possibility of big pollinators, or at least buzzing pollinators, getting to this area of the greenhouse. And still we see this huge fruit set. So this selfing mechanism makes sense for this species. And it might be the case that it's also for the other species, Arthrostemma ciliatum, as well. And this selfing mechanism I also found, just to go a little bit away, updates on other melastomes from the greenhouses here. In Calvoa pulcherrima, I found a huge mass of pollen tubes connecting the stigma and the anther. And in this case, it's actually a shower of pollen tubes. And I suspect the same with Calvoa, another species of Calvoa, Calvoa pulcherrima and Monolena primuliflora, because they also have this huge, insane amount of fruit set within the greenhouses without any, well, I don't know about the pollinators of this species yet, but there are definitely no bees going into the greenhouse, and the biggest insects are tiny, tiny gnats. And I suspect the same for other species of Calvoa and also Monolena, just because of the fruit set and the proximity of the anthers to the stigma. So coming back to Arthrostemma ciliatum, the two morphs I showed you, turns out, is not the whole story here. Recently, I went into iNaturalist and started looking at different uh, Arthrostemma ciliatum occurrences, and I was scoring them for isomorphic, dimorphic stamen morphs, just to see where the two morphs are co-occurring, whether there is hybridization going on, and came across this new kind of isomorphic form which has a longer style. And this was a completely new discovery for me, at least, looking at these pictures on a naturalist. And this started me to think about distyly, whether distyly might be happening as a pollinating mechanism here in Arthrostemma, where these two kinds of isomorphic and dimorphic stamen types are co-occurring in different places like Belize, some parts of Mexico, extremely abundant in Ecuador, and they might be just contrivance for cross-pollination. But there is also this form where the stigma never grows longer and it stays short, where in the isomorphic form. So we want to test this. At least we want to check the evolutionary relationships between these different morphs in Arthrostemma ciliatum, because this, as far as I know, is unique among melastomes, which shows these different style lengths and stamen lengths in the same species. And whether they are the same species is also a question that comes to mind. And recently, we ended up collaborating with a partner of ours here, a professor, Katia Romoleru in Ecuador, to plan field trips there, since it was very abundant. Both the morphs were very abundant there. And he sent us some pictures recently, which surprised me even more. The style that is longer, as you can see in this picture, is not always long. It begins as a short style and then it grows long later. And this was in the collected in the state of Pichincha in Ecuador. The style actually grows over time of the flower here. And you can see it on the same inflorescence as well here. This has a kind of a short style closer to the isomorphic stamen, but over time, this style grows longer. And in the same population, in the same areas, there is also the occurrence of this dimorphic stamen form. So it might, in Ecuador, it might actually be a case of growth of style and a kind of different kind of distyly going on here. So we want to also do a population level study in Ecuador to see what's going on with this. And I think it's high time to put these things into a phylogeny to see how this can happen within the same species, whether they are the same species. If the same species, maybe it's just undergoing speciation, or they are transitioning between different morphs. And it's all very interesting questions that are giving us a very nice future outlook to work on this genus. So yeah, plans. Future plans is check the phylogenetic relationships across the distributional range of this species. Also, maybe a phylogeny of the whole genus, including the different accessions or different morphs, to see their relationships with respect to each other. And we want to do a population level study in Ecuador, as I already said, which is looking quite promising now with this different discoveries every day. Of course, I want to go on a field trip to Ecuador as well to know these things in person. So far, I have not been to the neotropics. Well, the only field trip I've been to look at melastomes has been in India, where there's not many melastomes occurring, and to Cameroon. So it's a field trip, I think, to look at these things in the wild and to see what pollinators might be interacting with them is a very interesting prospect. Also, we want to do transcriptomics of all these three morphs, we get them into cultivation somehow, and check these genes again, and also, of course, check other groups in melastomes. And if you have any ideas, other ideas, suggestions for the same ideas that I've already talked about here, please do let me know. And that's the end of my presentation. I would like to thank many people, all the entire group here, which provide a very nice working environment. Gudrun, who came up with the topic and enticed me into this melastome world. Christian, who helped me a lot with the, the transcriptomic analysis. Katya, who helped with fieldwork in Ecuador and is helping and will help in the future. Anna Schlick, who did most of the pollination experiments with the pollen germination, which is surprisingly takes a lot of time. And these funding institutions for the funding, and that was it. That was all my work for the past 3 years I've been doing over my master, and I look forward to working on the same group for my PhD as well. Thanks. Hope I was on time.
Well, perfectly on time and perfect talk. Very, very impressive work. Thank you.
Very, very nice indeed. I have lots of questions. I'm sure that Agnes has a few, but so
I was looking forward to this discussion a lot.
and I'm
Should I
sure
stop
that
sharing now, or should I keep it?
you can stop,
Let's
and then
keep
if you
this.
need to show something again, you can.
Okay.
So, and I'm sure other people may have questions. So if you have a question, just unmute yourself and go ahead and ask your question. So I have questions about different parts here. That one of the first about the first part of the study. I was wondering if you looked into if there was any differential expression in matchbox genes, which have been associated with anther, because all of the other genes were different types, but like especially A or B class genes. I mean B or C class genes. I don't know.
Well, there weren't too many matchbox genes, but I did find one or two. But the counts were not really that promising to keep them as a candidate. I was also looking for those, which basically any genes that were involved in development, stamen development, flower development. But yeah, those might be the central coordinators of flower development. We already know that, and they might also be causing this differential elongation as one of the central transcription factors. But this might not show up in differential expression analyses, because usually these genes that are central genes, they are very sensitive to expression levels. Very slight changes in the expression of these genes can lead to a cascade of reactions, which leads to a bigger change in other genes. And this is why I was also talking about doing a network analysis with all the genes that I have, in order to narrow, or in order to make these into clusters, to correlate the genes that we did find as differentially expressed, to try and find what is making those genes differentially expressed, because these are not all of those are transcription factors, they are just transcripts. And to make transcripts, you need transcription factors, which are the central key regulators of most things here. So that might be the case, but in this, I didn't actually see any matchbox genes as differentially expressed.
I love the pollen shower evidence you got. I think this just your highlights the lack of natural history we still have for most of these groups. And I think almost every seminar, it doesn't matter if it's systematics, development,
Yeah.
population genetics that we give in here, that's sort of the last conclusion. So instead of putting it last, I want to put it first.
Yeah.
Here is how little natural history we have for a lot of these groups. And as soon as you had that short style, I was like, oh, that's probably the differentials, because they're really our concept of what is the pollinating and what is
Yeah.
not right here.
Yes.
But that's interesting, because in most cases, when you have those differential, the styles are long, but I see you have this situation here.
Yeah.
So I think it would be super cool if you go to Ecuador,
Yeah.
take advantage
Well,
of
Agnes and
yes,
also Katya
yeah. Well,
with their
let me
help,
know.
and also
I
Gudrun's
tag
help.
along.
Yeah, sure.
I have a lot of miles.
Yes, I will let you know.
One of the things you should definitely do is try to get as monochaetum on RNA later. And the reason I'm saying monochaetum is because in monochaetum is the opposite. Is the anti-sepalous stamens are small and the
Ah,
anti-sepalous stamens are long. And
okay,
that's
that's
sort of
interesting.
unusual. So most melostomes, the antisepalous are the larger ones, and monochaetum is completely the opposite. And in fact, there are even species with staminodia. So it would be great to have
Yeah.
the species with staminodia, the species with, and then the ones with good anthers. And then
Yeah.
there's even species of monochaetum with isometric stamens. So that I think would be another great group to look into,
Yeah.
just because of the comparison to Arthrostemma.
Yes.
And yeah, you and I could talk for 2 hours, so I'm gonna let other.
We can have a meeting of that kind soon.
Okay, sure.
Let me grab a pen here quick.
Okay. Other questions?
Well, I'll take the opportunity and jump right in. Fabian has already said things where I wanted to comment on as well. But like, if we start or stick with the Ecuador thing right now, so cool. That's really. I mean, yeah. I feel like whenever we start looking in the field, we just find more surprises. So it's
And
just about.
I've not even been to the field yet. I'm just looking
Yeah,
at pictures
exactly.
on the internet.
Wait for when you go to the field. You're gonna have 20 more.
Yeah.
But the two things that I think of now. One is right in that area in Pichincha where you found that species, you'd also have Mariania maxima growing abundantly, very easily accessible. That is also heteranthus.
Yeah.
I think if you put that on RNA later and look at it, you're probably going to find really different things, because the pedoconnective is not like, there's no elongated pedoconnective in that group in Mariania, but actually the appendage is really where the stamen dimorphism happens, with the appendage getting fat or small, changing color, and also anther length really changing. So that could be a very neat comparison for something that does something different if you want to get even more complex.
Yeah.
And also has close relatives that are isomorphic, so that could be pretty easy to sample. And looking at the photos, I was almost wondering if there could be, now thinking of Fabian's student Juan, who's working on Daisy, if what's going on there, it could not just be another case of a crazy progression into Daisy. No idea if that's very far-fetched, but who knows if pollen even is fertile in all of these small isomorphic.
Yeah. And in many cases, I've seen on pictures, especially in Ecuador and most of its distributional range, the stamen don't exist in the pictures. They have been bitten off by different wasps.
Yeah, or little trigona bees that munch up everything.
Yeah.
Yeah, very cool. And I also love your pollen shower observations. Honestly, now that I have more species in the greenhouse, I have also been noticing crazy fruit set. There are no pollinators.
Yeah.
I do 100% agree with you. Plus, so many of these smaller melostomes do close their petals. I think they even press the stamens against
Yes.
the stigma. That's
And
exactly what we saw in
this
Borneo. So,
this arthrostemma is surprising in that sense because it sheds the petals, doesn't keep the petals.
yeah, even
So
cooler.
it sheds it, and then I see pollen growing because
Mhm.
of the proximity. I think it's good enough for it.
Yeah, probably.
Yeah.
Yeah. And I also loved your thoughts on the constraints. I do agree.
Yeah.
I think spatial constraints are a big thing. So yeah, I'm wondering, I don't know, I mean, I think it is very hard to maybe even remove one world and see what the other world does if it has space to also
Yeah.
grow laterally. I don't know if that anything would happen or if everything is sort of constrained in the longitudinal axis.
Yeah.
But yeah, I think
But
you
that
have a
would be
treasure box to
that would
open.
be very difficult to very
Yeah.
difficult emasculation experiment to do with the
No, absolutely,
hyperthiums
with the
and
tiny bud. I know. But at least like a lot of, I agree. I've just looked at development in Vaccinea and I remember there that also there the differentiation and actually the appendage growth happens at the very end because there's just no space for two equally sized appendage whorls.
yeah.
That's another group actually where you have some sort of heteranthry, even though it's not functional in the strict sense of like feeding and pollinating anthers because
Yeah.
they're not depollinated.
Yeah.
So they have different reward type. But yeah, I'll stop here because there are other people with questions. I think we had one comment in the chat at least that I saw. Okay, that was Malu. She was wondering if the species could find a correlation among hybridization and apomixis in the Arthrostemma, I guess, in the field.
I have no idea right now. This is what we want to check as well with the population level in Ecuador, and I think we should check this across its whole distributional range, maybe different people in different areas. And the fruit set in Arthrostemma ciliatum, at least in the greenhouses here, and also from what I've seen in the pictures, is not that often. It doesn't happen that often. So I wouldn't think that there is much apomixis going on there, but I would definitely expect hybridization if these things are growing so close together with the two different morphs. I would definitely think that there is some hybridization going on there, unless these things are genetically isolated somehow. And there's also evidence for this because in Arthrostemma, the two morphs I showed you already, Frank Almeida has done a lot of work on this, you already know, and he suggested a correlation between karyotype morphologies and the stamen morphs. So there are two karyotype morphologies in Arthrostemma ciliatum. One is bimodal, where there's two groups of different sized chromosomes, and another one is unimodal. And the unimodal one is more associated with the isomorphic form, and the bimodal one is more associated with the dimorphic form. So whether these different chromosomal morphologies have anything to do, whether they make these populations completely isolated is a question to check again.
Yeah,
And
I was going. That was one of my other comments. Was, are these really different species? I mean, I think whether they're different, like, are the two things completely isolated reproductively or not? I mean,
yeah,
whether we call them species or not, that's a different question. But whether they're reproductively isolated,
isolated or not, yeah.
and Arthrostemma primavium, for example, we know it's tetraploid. It's not a diploid, and I
No,
wonder.
it's the reverse.
Oh, exactly. Sorry. Yeah.
Yeah,
So we have tetraploid and diploid species, but
yeah,
those are only based on literally like a handful of counts total, and
yeah.
most species never get counted several times. So these, I mean, counting the chromosomes and melanosomes is a pain in the neck, and
No, don't
doing.
tell me about it. I'm doing it right now, and
Okay. Well, I done quite a bit of it too. And flow cytometry, we have done it with fresh material and with very mixed results. we've done it with herbarium material and with very bad results. We've never been able to really do it well. Well, we doing the exact same protocol with grasses is worked perfectly.
Yes.
So
This is what I'm seeing with the chromosome staining as well. That it looks like they don't have any nuclei. I look at them, I stain them, I use a control as a monocot or even a dicot. I compare the melastome with the other one. The other one has brightly stained nuclei, the fulgen stain, Schiff's reagent, everything works. The melastome, zero, nothing. It's gray cells.
yeah.
The cells which seem to take up any stain look like pollen or pollen mother cells.
Yeah.
And I don't know, this is also very interesting for me because I come from a cytology background, kind of. I did cytology in my bachelor's. Yeah, this is a lot of interesting things in melastomes going on.
Yeah. So maybe
dig into this. We need work on that.
maybe some of the issues here is that tribe in particular, Rexy, we know that the DNA content is super low. I mean, we have grabbed species in the field, walk to the lab, and 20 minutes later do DNA isolations, and we get so little DNA per milligram of tissue. So I don't know if the cells are too big. That doesn't seem to be the case. I don't know if it is. I don't know what it is, but the amount of DNA we get is so low, and it's sort of common for all three genera,
Yeah.
for this
So
tribe.
that's bad news for my plants, sounds like.
Well, the
The three
advantage is
people
we know
here
we can
listening
grow
to the
them.
seminar, they all just started laughing.
Yeah. So I just want to say a couple people just joined recently. Maybe there was an issue with, and we have Constantine just coming in. There are issues with the time changes in the U.S. versus not, and in other places no time change, and other time changes earlier. So the seminar is actually sort of about to finish, but
Yeah.
we'll be putting it up, the recording, as soon as possible, and everybody can go.
Maybe I can share my screen again for my
Yes.
email so that if they have questions.
Yes, please do that. Any other questions,
Where's the
please?
share screen? Where's the share screen? Come on.
That's at the bottom of your screen, in the middle, green share. Yeah, there it is.
Yeah, so this is my email for anyone who wants to contact me. Well, let's go to the first slide. There's another one. There will be the recording, I think, online, like Fabian said. So, and you're welcome to send me emails. And more discussions would always be welcome.
I think you are starting on a very fertile ground. There's a job for a PhD, postdoc, and a career here.
Yeah, that's true.
Yeah. Well, if we don't have any more questions, it's already 1 minute past the time. Any more? One last question, or we can do it for later. Okay, well, thank you, Suvrat. Thank you, Agnes, for organizing this one.
Thanks for inviting me.
Yeah.
And thanks for joining. This was wonderful. A completely new topic for us. So it's wonderful to have.